Sweet. So here we go with our stuff, and we had left off yesterday with this idea that you had a couple things in the membrane. You had the proteins, you had the phospholipids, some cholesterol, some carbs, and everyone had a specific role. And now I think it's time for us to talk about movement across the membrane. So let's talk about membrane transport. So, what I'm going to draw out here is a makeshift cell. Assuming that this is an animal cell, because of the circular shape, certainly not a box shape, we could agree that this area on the outside here would be the ECM. What does ECM stand for? Matrix. So, this cell, if this was in your body, this could be one of your cells. Does one of your cells live independently?
No, sir.
No, it lives as a collective with every other cell that's in your body, right? Now, that means that we work together, right? So,
Why do you have a piece of thing?
it's just a long story.
Okay.
To bio,
Madame Hargis.
that was good.
Okay.
Very short, but to the point. I like it. So again, does your cell live independently? No. That means that the cells interact together. Now, if we interact, how do we interact? Do we stay on opposite ends of the planet if we interact? No. Well, nowadays we can, but yes. Because of the power of the internet and Google and all that other stuff, right?
Sorry, just had to be smart aleck.
No, that's cool. But I want us to certainly understand that for this to work out like we're hoping, cells have to be able to communicate. Now, cells don't talk because they don't have a mouth, right? Nor are they hooked up to the www, the World Wide Web, right? They can't Google each other. There's no Facebook on a cellular level, which means there has to be chemicals going out of a cell and chemicals going to the inside of a cell between that cell and the extracellular matrix. Now, you might say, well, where's this chemical going? Might be going to another cell. Albeit a cell that I'm drawing is quite far away, could be going to a different cell. Maybe there's a chemical coming from a different cell. But this communication is based on chemicals, which means we have to talk about how can the chemicals do this little piece right here. How can we describe this stuff right there, which would be the membrane movement or the membrane transport. So there's a couple different key terms that I want us to talk about. So here's the first key term. Diffusion. Diffusion. It's basically a tendency for molecules
Oh, you things fixed. No.
no luck, huh?
Yeah, we need to just why do you have a piece of paper in your hair?
It's tape. My hair was sticking out. People were being very rude, so I put tape on my head. Nope. I'm actually hurt. I'll be okay.
We all love you. I'll make you some brownies. We're all like your
Do you want some cookies?
No, I'm fine, I'm fine. I really just need a haircut.
Here's a lot of cookies.
Still up?
Can I have
And I don't like oatmeal raisins.
Oh my gosh, I love oatmeal
Do you
raisin.
want some?
I'm fine, thank you though.
It'll make you feel better.
No, it won't. Okay, so diffusion, the ability of molecules to spread out evenly across a certain area. So, now there's just the logic here. Let's imagine something real quick. Let me draw out a picture of my room. So let's just assume that this is my room. And I'm going to let these little orange dots represent people. Is it logical that everyone stays in one corner of the room? They want to maximize space most likely. So what will happen is one will go over to here, and one will go over to here, and over to here, and over to here. And that's one thing we see is that by the time this is all said and done, really their placement is more spread out than it is anything else. Cool. I didn't mean to make the room bigger. I just mean to make the particles more spread apart. My bad. And so we say that what's happened here is we've reached something called dynamic equilibrium. And we've talked about dynamic equilibrium before with chemical reactions. It was a case in which you have still molecular movement, but an evenness or an equal movement to the left and to the right. So movement still occurs, but particles maintain their max distance.
Now, why do the particles just want to do that? Do they just want to?
It's really because the repulsions that are like particles are made of atoms, right? Maybe the particle's a compound, maybe it's just a single element, but certainly it's made of atoms, right? Van der Waals forces basically state that atoms dislike each other, like they dislike each other, and if that's the case, then they would push each other apart, and that's what we're seeing here. So I've got a fun-filled, action-packed picture for you. I'm going to show it to you. And I was hoping we could either get an I agree or I disagree with what this picture's saying. They're all on the left side. Some start moving to the right side. You can see in that middle picture. And then equilibrium. See how they're about even on both sides? But are they still moving? A dot from the left is moving to the right. A dot from the right is moving to the left. So equilibrium doesn't mean like we don't change or we don't move anymore. At equilibrium, they still move, but they move equal ways. In other words, this one goes to the right, one goes to the left. If 5 went to the right, 5 would go to the left. Cool? Awesome. That's fun. Substances tend to travel down their concentration gradient, which means they basically go from an area of high concentration to low concentration. Because you're going down your concentration gradient, you're going from where you are a lot of to where you are very little of. We say that this movement down the concentration gradient is a passive process, which means that there's no need for energy input. No need for energy input. Now there are two types of diffusion that I want to talk about. So let's kind of zone into types of diffusion. We good? What are the two types?
Hypertonic and hypotonic.
That would be like relative concentrations. I'm thinking of one that involves a protein and one that doesn't involve a protein. Do what? Sorry, I just lost my train of thought. atoms are so close. Cool? So, facilitated diffusion. What's the difference between simple versus facilitated? Let's look at this word facilitated. What is that kind of standing for? Where are we going with that?
Somebody has to make it.
Someone's helping, right? Someone's facilitating. Like, who's the facilitator of biology?
Proteins.
Okay. So in the case of facilitated diffusion, you have a protein that allows the movement. In simple diffusion, the solute particle can simply move through the membrane. Now, we talked about only a certain small group of molecules being able to pass through a membrane. What were those molecules?
Very tiny.
Very small, very nonpolar, right? So the only thing that can do simple diffusion is basically something that's very small and nonpolar. Those were two great examples, the two best examples.
What does it say?
Ear infection is not like.
Do it.
What does that last line say?
Membrane.
Okay, but like the whole one.
The solute particle can simply, simply go through the membrane. My apologies.
When's the last time you got a haircut?
The last time I got a haircut? I don't know.
10 years ago.
No, let's be realistic. I mean, it is like maybe 3-4 weeks ago.
Not sure if Cal ever on the tape.
It's.
I'm sure it's down.
I'm not too worried about it. You know, in the end, are we okay with how the difference between these two though? Facilitated, protein's got to let it through. Simple, no protein at all. Do they require energy?
No.
So the facilitating, like I guess adjective there maybe it's an adjective. I don't know, but or adverb. I don't know. But this term is like protein based, right? Protein helping. Right? Okay, sweet. Now the next thing I want to talk about is I want to talk about a special type of facilitated diffusion. So this is like a special feature. And that's going to be osmosis. Don't say Jones. It's just osmosis.
I saw it really recently.
I think we all
And it was
think
still-
it's a show too.
Yeah, I like the
What's
movie
that
show.
song?
Yeah.
I think it's an American classic.
It's
It's a movie TV show. If you have Netflix, look it up.
I don't have that.
They had it's basically
Yes.
like a
Yeah, I think
buddy
so.
cop
I think
movie
I
based in the
most
body, like a white blood cell and
or
pill work
most
together
is
to
the,
bring down this deadly virus.
That's on
And here's
Umbrella
Chris Rock.
Rose. It's really good.
Voice is awesome, Mrs. Jones. It's Chris Rock.
Yes.
I don't know. I never saw it.
You have to. You're a biology teacher.
Well, I actually
Definitely wasn't a requirement for my degree.
Nowadays it is.
Nope.
Please.
That wasn't part of the curriculum
Can we
today.
bring
No.
it in after the AP test?
Probably not. We
We
have.
should all just get together and watch it, just like outside of school, you know?
You have to invite Mr. Fields too.
No, I can't watch programs like that. But osmosis is the movement of water, right? Only the movement of water. Didn't say a solute particle. Not talking about dots here that are moving. I'm talking about water moving with relation to the dots. And so the way that it works out, and this becomes a little wordy, but water moves. I'm fine. From an area of low solute concentration to high solute concentration.
You look like a turkey.
Want to make sure that we get that we're not talking about we're not talking about dots moving, we're talking about water moving here.
So it's just to make it like more spread out, like.
It evens out the dot distribution, but not by moving the dots, by moving the water.
Yeah.
So to help us with that, I've got a fun-filled action-packed slide to show you, or a picture from a slide.
Yay.
Thank you. So this one right here, this is YouTube, not the website, the real thing. That joke was so much better like 3 years ago. That's cool. So if you look at YouTube, you see the left YouTube? The water levels are the same, right? Talk to me about dot distribution though. A little bit higher on the right-hand side over here, right? Now, in simple diffusion or in facilitated diffusion, the water levels would be maintained at the end, but the green dots would move to your left. Are you seeing that in this case? No, what's moving to your right? Water. Water went from an area of low dot concentration to high dot concentration. See, water went this direction, and we see that over here. That means that this membrane right here is selectively permeable to water and water only. That means like the particle would be, or the dot particle, the solute particle, would be too large to pass through that opening. Only water is small enough to pass through that opening. Do you understand?
Yes.
You can even see that when they zoom in down here. What do we think? This is fun. Perfect.
So how did so it evens out because it adds more water to it, thus giving it more area to disperse?
Yes. Yes. So it moves the water to reach an equilibrium. Not the dot though. Diffusion up here, the facilitate simple, that was dot movement. That was solute particle movement. This is water movement. Some terms that we need to know that's associated with osmosis. The term isotonic, hypertonic, and hypotonic. In the case of isotonic, that's always the easy one because this is when you have equilibrium. You have an equalness of stuff. You have solutes, you could say, on both left and right sides or on top and bottom, wherever you're talking about. Now, in hypertonic, when someone says that this guy's hyper, in your mind you're just like, that's too much, right? So hypertonic is always referring to too much solute. And hypotonic is referring to too little solute. So we have these different types of solute particles. So I have a fun-filled, action-packed picture to show you now.
Another one? Gosh, you're so generous today.
I really kind
How
of.
many videos?
A lot of
The videos come later.
You promise?
Nope. Maybe. Depends.
It's too ambiguous. I need a concrete.
You need concrete answers.
Would you sign a binding contract for that?
No.
What?
On an unrelated note, could we watch the Magic School Bus after the AP test?
No.
That's so far away.
What's your plan after the AP?
Study.
Study for next year? Or just study?
Like, what will we study? We're all going
Probably
separate
molecular
ways.
biology.
Study what?
Molecular biology.
Can't we breed animals?
Can we what?
Breed animals?
We don't have that kind of time.
We don't have
Mr.
that,
Fields,
so-
can we get a class pet?
We could have, but no one ever got one.
I have a guinea pig. Can I bring it?
It's sugar better.
There's no way I could take care of a guinea pig. I can barely take care of myself. Look at my hair.
I have a brother.
We can't. We obviously can't use your brother as the class pet.
He's
Why not?
really close to being a monkey, so I'm sure we can do like studies on
What
him.
are you saying?
Okay, we'll consider that. Okay, so what I need you to do real quick is we're gonna take a couple seconds and we're gonna get everyone to copy this little picture down, and then we'll discuss it. Going to the Kroger, the grocery store, you go by the vegetable section. They always have the little water sprinkler systems on the lettuce, don't they? If they put a ton of water around the lettuce is going to be forced to take a bunch in. That causes the lettuce to have a turgid appearance, and you hence buy it. If they didn't put the water out on the lettuce, water would leave the lettuce excessively. The lettuce would look like plasmalized, and no one would ever buy it. It's a marketing technique, right? Now let's talk about our key terms, the hyper versus hypotonic. Are you ready for this?
Yeah.
Water seems to be going to the inside. Can we agree on this first part right there? The only way water would ever go inside is if there was more business on the inside, more dots on the inside. Agreed? Water would only go in if there was a lot of stuff on the inside. So I would say that this solution, the solution is going to be hypotonic.
Is the solution on the outside of it?
Mm-hmm. Now, what if I was to say the cell, though? What would the cell be?
Hypertonic.
Hypertonic cell. Do you understand why it's hypertonic? Why the cell's hypertonic?
Yes, sir.
The cell has a lot of stuff on the inside. That's why it goes to the inside. How about in the case way on the far right? Water appears to be doing an exit from the cell. That means there would have to be a lot of dots on the outside of the cell. Agreed? There'd have to be a lot of stuff on the outside. So if stuff was to leave, or if water was to leave the cell, then that would mean that it's leaving because the solution's got to be hypertonic.
So if is there ever going to be like simple what is this like hypertonic or something? How would you answer that
It
in
de-
terms of like cell or in terms of
It depends. They would say the solution is blank with respect to the cell. So in this case, the solution is hypertonic with respect to the cell, or it's hypotonic. Or then what if they said the cell is blank to the solution? The cell is hypertonic to the solution. So the terms hypotonic and hypertonic are directional, if that makes sense. It's based on like location, where your description is from. Cool? So that's all about osmosis, right? Now, I had said before that osmosis was a special type of facilitated diffusion. Now, if it's a special type of facilitated diffusion, that means someone's got to be facilitating it. Which, and we could guess, it's certainly going to be a protein that's facilitating it, right? Yes, definitely. The name of that protein is called the aquaporin. This is the protein that allows water movement. Yep.
Did you take off the tape?
so that
Okay, because I didn't see it. It
it's
looks
just
better.
getting like is your hair wet?
It's
It's
gel.
If I don't, it goes everywhere.
Can you do that one day?
No.
Please.
You probably look like Ted Mosby.
I don't know who that is.
Yes.
So, the last thing I want to mention is I want to mention a certain type or another type of facilitated diffusion called an ion-gated channel. What an ion-gated channel is it's basically a protein that can allow an ion in or out of a cell.
Should I start a new page or should I?
I would keep writing.
But I have three lines left.
That we're about done here.
I'm OCD about my notes.
Everything we've mentioned thus far, including osmosis, was a passive process, one that did not involve an input of energy, right? So let's switch our focus real quick. And let's talk about a different type of transport. Let's talk a little bit about active transport. Okay, obviously active transport is going to be using some energy. Cellular energy is ATP. We mentioned that before. And what it does is it allows for solute particles to cross the membrane. And as they do so, they're going up their concentration gradient. So a good example, or I'm sorry, a good way to view this would be, imagine that this is my box again. This is my semi-permeable membrane, my SPM as I'm calling it. Put all my little dots over here. Dots are also the solute particle. Dots are also a great candy around Halloween too.
What are?
Dots.
They're they used to be my favorite, and then the same. Sorry.
It's good. Dots are good for you.
I know they're rich when they give out like king size during Halloween.
Or when they give out apples. Seriously, one time someone gave us a cupcake. I was like, a homemade cupcake. They're like, don't worry, there are no razors in it. we didn't eat them. We probably threw them out on the street.
Notice my arrow's going to the left side now. The side with few dots is able to facilitate those few dots going to a side that is heavy in dots. It's like taking a room full of people. Imagine there's 500 people in this room, and us trying to stick 100 more in it.
Yeah, it's the light.
Like right now.
And he had to use energy to get in the door too.
Is Halloween on a weekday again this year?
I think it's on the Tuesday.
It's so awkward.
Is that? I don't know.
Goal with active transport?
What would be the purpose of it?
It would fire a neuron, and you're basically sending electrical impulse throughout the course of a neuron, which is a nervous cell. You have to have a ton of sodium and potassium change sides. Basically, sodium moves from an area of very high to very low concentration, and potassium moves from very high to very low concentration. Well, that would allow the firing of a neuron one time, but then you have to be able to fire the neuron again, so you got to replace all your ions back in the right spot. And an ATP active transport system would allow you to, if you will, fill a room up with 500 people so that the room essentially explodes. And I needed to explode again, so I have to refill it with energy up to 500 people. And that's actually the way your nervous system works, which explains why you have to have food to run like just the basic components of your life. Thinking requires energy. It's pretty cool. A couple other little pieces that I want to go through, and then we can get close to being done. First off, it's something called electrochemical gradient. An electrochemical gradient is basically when when the solute particle is a charged particle. For instance, there's going to be a time and place where I draw out a structure like this. Do you know what I'm drawing out right here?
Mitochondria.
Perfect. And what we're going to do at some point in time is we're going to show you how there's a bunch of little H plus protons, this is called, some hydrogen ions, a bunch of little H pluses to the outside here. Now, there's a lot of H pluses on the outside here. There's very few in the mitochondrial matrix. So the H pluses definitely want to go through and go down this direction. Agreed? And there is a facilitating protein that allows that movement. Do you see that in brown here I've got my facilitating protein that's going to allow the movement of my H pluses from the inner membrane space to the matrix? This gradient that you see right here is a gradient that is an electrochemical gradient.
These
Thank
two
you.
I don't know what to do.
Awesome. Thank you. This right here, that's like a protein that's allowing this facilitating the movement of H pluses. Is it going to be a passive or an active process? Passive, because the H plus is going from an area of lots to an area of littles. Now let's take that electrochemical term apart. What do you think that's referring to? There's a lot of positive on the space in between the two membranes, and there's not a lot of charge on the inside here. So there's the electrode difference. Positive wants to go to where negative is. That's one thing. Chemical is like a particle, right? There's a lot of H plus over here. There's no H plus on the inside. So for two reasons, the H plus wants to move to the interior. The electrical reason and the chemical reason. So the H plus goes through this doorway that is this protein that looks like a bell almost. It goes through it with a lot of force. We'll talk more about that soon. But do we at least understand the idea of electrochemical gradients?
So the hydrogen that's positive becomes negative.
I'm sorry, what? Say that again?
I'm not reading
Nope,
that.
no. What I'm writing over here
Uh-huh.
is I'm writing that there's a lot of H plus on the outside and there's very little H plus on the inside. So I'm talking about concentration here.
Oh, okay.
There's tons of H plus, not much H plus in here. So the H plus for just particle wise, for chemical wise, wants to go inside. Now for electrical wise, it wants to go inside as well. So you have two reasons that this H plus is going to move inside very quickly through this one hole.
Then why does it need protein?
Because the deal is that H plus is a charged particle, and charged particles typically can't get through the nonpolar hydrophobic region of the membrane. So they have to have a facilitating protein to allow their movement. And also, having a protein facilitating it allows a protein to regulate the process as well. As we come to the conclusion for today, we've got one other type of transport that I want to put some focus into, and that's a type of transport called bulk transport. We think about bulk transport, we're thinking of the moving of large items, right? So this is going to be the movement of large items. And typically this is going to be energy requiring. And that's because you're talking about the movement of very large items. I don't know why I drew that. I just felt like doing that, I think.
I was about to ask about that.
I have no idea what's going on right now.
I like your underline. It's a really elegant one.
Oh, thank you. Yeah, that's really nice, isn't it? Let's talk about... No, eventually. Let's talk about two types of bulk transport. Let's talk about endocytosis. And let's talk about exocytosis. And if we use our prefixes to our advantage, we know that the site in the middle there means cell-based. Endo means to come in, exo means to go out. So in endocytosis, you have incoming bulky particles.
What would be an example of a really bulky particle?
It's really not bulky particles. What happened?
That's my mom too, Corinne.
Corinne's and your mom.
That's so mean.
So uncalled for.
It's really not that there's a large particle. You're taking in many of just a decent sized particle.
Okay.
So it's like, it's not saying that the piece of trash is very large or the food particle that's coming in is very large. It's you have a lot of it. As opposed to exocytosis, which is when you're having a ton of outgoing or exportation. Bulky particle movement. So we're definitely getting this idea that things are coming in, things are going out, And we're not talking about necessarily that it's a large item. It could just be a lot of that same large item or medium-sized item. I want to put some extra focus in endocytosis, and I want to talk about a couple of specific types. Phagocytosis is one of them. There's also pinocytosis. And lastly, receptor-mediated. Endocytosis. In the case of phagocytosis, what we see here is this is when the cell basically is eating stuff, taking in food items. In pinocytosis, this is basically like a cellular drinking. In the case of receptor-mediated endocytosis, this is a case where you have something being taken in by a cell, but it's taken in by a protein that is stimulated by a ligand. A ligand, a chemical.
What's with that name on that last one?
Chemical enhanced endocytosis.
What's the difference between cellular eating and cellular dining?
Drinking.
Oh.
Eating is going to be when you're taking in like proteins, carbs, lipids, all that stuff. The pinocytosis is where it just engulfs it's like some of the fluid from the ECM, which might contain dissolved of the things that it eats, but it's not specifically like food particles. I have some fun-filled videos to show you real quick. The one though, I'm going to choose the one that I think is the most important because it's the one that's the most complex, and that's going to be the one over receptor-mediated endocytosis.
Unlike phagocytosis and
So,
pinocytosis.
hold on real quick before you leave. One little video, then you guys can jet. Actually, tell you what, we'll watch this video tomorrow. It's cool. Y'all can go and go. Thank y'all very much.